Hello, everyone. Welcome back. I hope you had a good break. And we are now going to have the last few talks of the conference. So it is my great pleasure to introduce John Murray from the Department of Genetics at the School of Medicine at University of Pennsylvania, and also has the lab next door to mine. So anyway, take it away, John.
Thank you so much, Arjun, and thanks to both you and Shalav for the introduction or the invitation to speak at this amazing conference. It's really just been a fantastic few days for me. Every talk I feel like has some method or biological or theoretical insight that's directly relevant to the work we do. So just kudos for this amazing conference. So what I want to talk today about is development. And we've had a number of talks at this conference about development. And typically, people start off with a slide like this, where in a single cell context, we're really thinking about trying to deconvolve the complexity of this incredibly complicated process, right? So you see a lot of different things happening in these different embryos as they develop. Cells are dividing, they're moving around. There's adopting different sizes. Some of them are turning fluorescent. And they're doing these all at very precise times as well. And so I'm going to tell you a little bit in the beginning about some of our work to map gene expression across development in this organism in the top right corner here, the C. elegans embryo. I'll tell you why we think that's a great model in a second. And then leave a little bit of time at the end to talk a little bit about the temporal control as well about development. So why is C. elegans a great system to apply and sort of develop single cell methods in addition to answer interesting biological questions? We're certainly very interested in the conserved mechanisms. So obviously C. elegans, and while it's small and transparent and develops very quickly, it has muscle cells and neurons and all the basic cell types that we know and love from more complex mammalian systems. Perhaps more real critically important, though, is it has this really robust mode of development. And so back in the 80s, John Sulston mapped out this entire pattern of cell divisions that each C. elegans embryo goes through as it's developing from the zygote to ultimately a worm that swims away. And what's unique about this system is it's incredibly robust. And so, we saw a lot of examples of developmental robustness in other organisms throughout this talk. In C. elegans, the actual pattern of cell divisions in every wild-type individual is invariant. And so you can look at any given individual and follow a particular pattern of cell divisions. We know what that cell is going to become in terms of its cell fate. So how do we take advantage of this invariant lineage to map gene expression across the lineage? We've seen, again, in this conference, many examples of the complementary advantages of imaging-based approaches and sequencing-based approaches. So you can see in this movie that's developed, that's going along the left side of the screen here, as the embryo develops, we can really see which cells are dividing and how they're all related to each other. If we follow this movie very carefully, and we could actually follow from the zygote all the way to the terminally differentiating cells. What are the pattern of cell divisions that led to that? And with very high certainty, right? And we can see, some cells maybe will be fluorescent. We get very high temporal resolution from that as well. You can see exactly when, particular reporter gene comes on in this case. However, the throughput of that is much lower than in terms of the genome than say sequencing-based approach, where we can do large-scale sequencing. We can get information genome-wide about how different genes are expressed across cells. What's missing in the sequencing case, and the previous talks notwithstanding, amazing approaches that are coming online to try to get around this, but it's basically a static snapshot, right? So each of these dots in this case, the UMAP projection of a single cell data set is a single cell at a single time, and we have to infer based on something about the pattern of trajectory, the similarity of cells that form these trajectories, similar states, that there's some lineage relationship there. Right, so I'm going to start with a little bit of the background on the imaging approaches. We basically, our goal when I started my, getting into the system back in the early 2000s now, was to use this as a system where we could really think comprehensively about mapping gene expression, both for every gene as well as in every cell of the organism. And because of this invariant lineage, we can actually make a list of all of the cells of C. elegans at any given stage. And then, if we can figure out the expression of each of those, we have, in principle, a complete description of for example, mRNA expression across development. Right. So using a movie like the one I showed you on the last slide, we can use cell tracking software, the program called Starry Night that was developed by Zhurong Bao and I back when we were postdocs, to track the cells across time through cell divisions, quantify gene expressions, noted by these funny yellow basketball-looking object here, to account for liquid background correction. And the output of that is the lineage tree of that particular embryo that we watched the movie for. That's denoted here in this purple color. And we can quantify in each cell at each minute throughout development from these movies the expression of a second color fluorescent reporter. In this case, it's a GFP fused to this transcription factor FA4, which is a conserved master regulator of gut development across all animals. Right. And so this is a nice lineage tree. It shows you, even for presumably most of the audience isn't the expert on C. elegans lineage development like I am, but now if I told you that this gene is expressed only in gut cells, you have some sense of where in the lineage the gut comes from. You can see there's several different clones that produce gut cells. Some of them are larger, some of them are smaller. This one on the right here is the intestine. The little small ones are the rectum. The rest of that is sort of the more complicated pharyngeal apparatus that is involved in eating and swallowing food. And so it's a single cell resolution, high temporal resolution map of gene expression for that particular reporter. And the great thing about this, we just turn the crank over and over again for many different reporter genes. And so here's an example of just 4 different genes that are, again, conserved regulators in different tissues: muscle, gut, intestine, gut more broadly, and neuron development. And because it's the same lineage every time, we can say that this cell in this embryo that's a MyoD expressing embryo is the same cell essentially as the cell from this N3 expressing, or this cell from this FOX4 expressing embryo. And we can map those all onto a single reference lineage now, where we have the expression of these hundreds of genes all mapped to the organism at single cell resolution. And that's in this database, that EPIC database. Expression patterns of neurocytes, not a medical record database. Right, so switching gears to sequencing studies, right? This has just been an amazing explosion the last few years in terms of the ability to map gene expression across entire developing embryos and time courses. This is an example from zebrafish that I think we saw in several previous talks. Looking at going from the very earliest uncommitted states in different tissue and germ layers. And the basic principle for these approaches, as probably most of you are familiar, is that you have some different stages of embryos. Maybe you have them stratified in different wells. Maybe they're mixed together. You separate those into individual cells. You run them through something like a 10x or a drop-seq or an indrops, or some other single cell approach, and then you get transcriptomes for them. Obviously, they're very sparse transcriptomes. And the hope is that there'll be enough information there to be able to assign that cell to its appropriate place in the developmental trajectory. And in most organisms, this is a discovery operation, right? Where we actually, we know the basic broad brushstrokes of how cells are committed to different fates. We don't necessarily know all the details. In C. elegans, that's not true. We actually know the right answer for this. So this is a great test case to see how well we can do for this, right? And this is work. There was a great collaboration with Jonathan Packer, who was a grad student in Bob Watterson's lab and co-advised by Cole Trapnell, and Chen Zhu, who's a grad student in Jin-Young Kim's lab in our biology department at Penn, right? So I'll just start this manually. So we did this. I'm going to go through this atlas relatively quickly because it's published, and just highlight the key methods and a few of the, I think, interesting insights that we got from this approach. This is an atlas of the C. elegans embryo based on about 100,000 cells from all different stages. And this is a 3D UMAP projection of those cells. And they're color-coded by, you can think of that as basically developmental stage. So the green cells are early progenitor cells, and as they go up to blue, and then red different tips of these different branches. That's the different cell face radiating out, and as the cells develop. And I don't want to dwell on it too much, but basically, it turns out, and then we did a lot of work downstream to validate this, that we can take mixed stage embryos and then figure out after the fact what stage embryo each cell came from by basically mapping the cell's transcriptome to a whole embryo, really high resolution whole embryo time course data. And this on the right is just a validation of that, where if we age the embryos, we lose the early cells, one of several validations that we did. So we know for each cell what time embryo it corresponds to. And obviously, like people typically do in these atlases, we went through and used marker genes to annotate different tissues and cell types, right? So this is looking at the entire data set. You can see all the major tissues, muscle, skin, different types of neurons, intestine, and so forth, as well as more specialized cell types. If you're C. elegans cell type wonk of some kind, comes, oh, I'm really interested in the head mesodermal cell, and I can say, oh yeah, we have the head mesodermal cells. We have basically every specialized cell type, that we can, with very different cell type, we can identify them in these. And this whole organism level view doesn't give you a totally resolved picture of all the cell types. And this is something I think has been hinted at a few times. Just want to emphasize it. You really have to do some sort of iterative analysis to try to tease apart more specialized cell types. So for example, zooming in on this group of cells, which is the ciliated neurons, the sensory neurons, if you reproject those, now you can get a much higher resolution map, where we can go through again using literature markers, and each of these terminal branches is a neuron, and we can identify each of those. And basically, this is all of the ciliated neurons that are present in the embryo, which is pretty cool. There's a total of 20 born in embryogenesis. I think there's one that's not on the diagram, somewhere else in the plot. So that's great. We have this, like I said, we know the right answer, and basically we can identify all the cells we expected, and we don't find any cells that we didn't expect to find, which is also sort of nice. And on top of that, we can start seeing some hint that maybe we can also identify progenitor and lineage relationships. And so in this area that has this mark here with these black hash marks, there's a group of cells, the ASJ and AUA neuron, for example, which appear to branch from some common branch point. And in that case, that actually is what happens in real life. They actually share a common progenitor, and that progenitor is then the sister of a progenitor for another neuron called ASC. You can see that branch as well. So that's all good. But then there's other stuff here that doesn't make sense, right? So for example, the ASE neuron itself actually is really two cells. There's a left and a right. And most of the cells in the embryo are left-right bilaterally symmetric, and we don't resolve them. We basically see, ASH neuron, for example, there's two of those, and we just see them all pooled together as ASH neurons. But for ASE, after it's born, they actually become molecularly distinct, the left and the right version. And the ASE left neuron is actually over here. Like it totally teleported to some randomly different place of this projection. So again, like those of you who work with these projections a lot know that, this is maybe not too unexpected, but this kind of thing happens, and it's certainly be cautious about over-interpreting proximity in these kinds of diagrams. Right. So just taking another step towards the early annotations, we can basically do the same kind of annotation, looking at these different projections for cells that just came from early embryos, and using the imaging data that I showed you before to annotate them. So a schematic of how this works. We have this, various genes whose expression pattern we've mapped by lineaging, like the STAT51, MS lineage, and then only a subset of the cells that are derived from that lineage express HAND1 or 39 genes. And so we can combine these to get very high resolution pictures of which cells come from which lineages. And this is sort of overwhelming view of sort of what some of that annotation looks like. I just want to highlight this one point from here again about the sort of where the projections can lead you astray. So just zooming in on one small part of this over here, you can see that there is this branching pattern again, and which the tips are correct, right? So the red group and the red cells here and the blue cells, those correspond to the red cells and blue cells up in this schematic of the true lineage on top. But actually, the next branch here, these are not sister cells; these are actually cousin cells in the real lineage, and the sisters of these are actually in some other part of the plot. So again, with a high resolution. we're able to tease this apart in Cellular because we have all of these markers. I think this really emphasizes the value of some of these experimental barcoding and lineage tracing approaches that we've seen in some of the other talks of this session. So at the end of the day, we have the entire lineage essentially, well over 90% of the cells covered by the single cell RNA-seq data, where we can, and the coverage is pretty high. We have hundreds of cells from most of these branches, and so we're able to get pretty high accuracy quantifications of transcripts for all but the most lowly expressed genes. Just an example showing RNA versus protein for this one for gene that we looked at earlier. So that's great. Like what have we learned from doing all this? I'm going to highlight just a couple of examples of things we emphasize in the paper, and then move on to talking about timing for the last few minutes. So one thing is that there's a lot of examples where cells from different lineages converge to the same fate, and actually that's pretty common. And molecularly, we appear to nearly completely lose the lineage history when that happens. So the cells become indistinguishable. You can't see their lineage markers anymore after they come together. There's a lot of examples of what's called multi-lineage priming, where you have a regulator of a particular cell fate that's actually expressed in the parent before that cell is fated, and then it disappears in the sister of the cell where that regulator is not affected. Early on in development, most of the transcriptome changes we see are really highly correlated with lineage identity, but then there's a rapid transition when cells start to differentiate, where often there's a large jump in the UMAPs as well, and the cells switch from the transcriptome changes being much more correlated with their terminal fate. This is just an example of some of these jumps, and we've seen a couple of those already. For example, this IL socket type 3 parent cell, you see one of its daughters is a type 3 cell, which is contiguous, but then the other cell is in a totally different place in the UMAP. And we can actually quantify not just looking at the UMAP, which can mislead you, but looking at the levels of gene expression differences that have occurred in these different branches. And actually these teleporting branches actually have more quantitatively more changes in gene expression on average. All right, for the last few minutes, I want to talk about how this project has helped us think a little bit about really a fundamental question in embryonic development, which is how you achieve precision in gene expression. And I told you already that it's an incredibly robust process, the same pattern of cell divisions in every animal. So that's going to require spatial precision in terms of expressing regulators in the right cells, temporal precision to express them at the right time, and also dosage precision to express them at the right level. And actually, Arjun's postdoctoral work was sort of like some of the most beautiful work showing how important this can be, where basically the idea is to specify a particular fate, you need to increase the levels of a particular regulator to a particular threshold level so it can induce that fate robustly. And it has to happen during the right time window as well. In the case of C. elegans embryos, it's happening in the context of like a 15-minute cell cycle in some cases. Right, and this precision can come from a lot of different mechanisms, right? So like in the fly embryo, and the blastoderm and other tissues like the C. elegans germline is another example of this. We take advantage of spatial averaging in syncytial tissues, where individual noisy nuclei can share transcripts across the cytoplasm. In slower developing systems, you can have temporal averaging and just rely on a long time for transcripts to accumulate and achieve some steady state level. One thing I think that can be very widely used and maybe discussed a little bit less often, but is probably the most important mechanism in developmental context is redundancy. That's both things like redundant enhancers, shadow enhancers, or so as they're called sometimes, or even like redundant paralog regulators that basically have the same function and sort of can buffer each other's noise. And so in the C. elegans embryo, and this is what Arjun's work showed, and we got into this actually through a collaboration with Arjun. We got to Air's beautiful 2013 paper, showed that it involved, so Arjun's work involved the robustness of the specification of intestinal fate, and this lineage is called E. And basically, if you look across expression across time for the key regulators of this, there's these genes called N3 and N1 and L2, and N3 and N1 turn on L2, and L2 is the master regulator of gut specification, and they have these thresholds in terms of making gut specification. But what I want to highlight is something a little bit different, which is the incredibly high levels that these genes go to. So N3, for example, goes from 0 to about 600 mRNAs in a single cell in about 15 minutes, which is this really impressive rate of transcription. Right. So we looked at the single cell data, and this is actually looking at a different data set than ours that was focused more on the early embryo, but the same basic logic applies actually to the analysis of ours as well, and see similar things. And so basically the idea here is, can we learn something about absolute levels of transcripts from single cell data? Which we know there's biases from sequencing. So is this accurate? So we went ahead and we just calculated what are the number of UMI or number of reads, depending on the data set in a mother cell compared to the daughter cell, and use that to infer how much new transcript has been produced in that one cell division, and use a series of cutoffs to basically find genes that we thought were expressed at exceptionally high levels. You can see that at 4 cell stage, there's very few of these, but by the time you get to 8 or 16 cell stage, you actually have hundreds of genes that are expressed at this level where they're increasing by more than 100 transcripts in these very short cell cycles. And so we did some single molecule RNA fish, again, taking advantage of Arjun's lab and their expertise and their microscopes. And this is some examples. You can see like really high spot counts for these genes, like X35 or C51, which are all conserved transcription factors, as well as being expressed at very high levels. They're involved in different lineages, state specification. Right. So overall, if you look at the maximum level measured by RNA fish versus by single cell RNA-seq, the correlation isn't perfect, but it's actually it's very consistent with what we saw. With again, an earlier collaboration with Arjun looking at melanoma cells, where basically you're able to predict the absolute levels probably plus or minus twofold or so. And so we think that's enough to identify these very high rate genes is a pretty accurate proposition, right? So I just want to emphasize, how high these levels are, right? So 2 examples in a particular lineage, TBX35 and C51 is the parent, this is the daughter cell of this MS division, right? And so C51, as an example, goes to almost 1,400 molecules in again, it's like 20-minute cell cycle. So these are accumulation rates of dozens of transcripts per minute. You're essentially packing the polymerase, almost as close together as you can if you assume in vitro transcriptional elongation rates. So I'm not going to go through the details of it, but basically Priya Ramakrishnan, who's been doing this work in the lab, has done a lot of dissection of using this list of genes that we identified from the single cell data to identify features of these. This is one example where one of the features she identified is these are high rate genes are enriched for this INR promoter element. And if you mutate this element in the C51 promoter, you can see you decrease expression by almost half. And sort of long story short is that we think that these genes, there's not like one specific mechanism that controls this really high rate, but basically all of these genes have a number of different promoter elements that combine to give you this exceptionally high rate of transcription. And so this is sort of finishing with a model for why we're seeing these really high rates of transcription going on. So what's happening in the C. elegans embryo, you have, I said before, it's like a 15, 20-minute cell cycle. You have to make a decision. And then to become a particular cell fate, cell's going to divide and have to make new decisions. So you really need the protein level for the regulators to have a step function like pattern of change, right? And the way you achieve that is to have something like this for the mRNA, where you have this incredibly high spike that's very transient and then goes away. And actually, we can see this offset here, going back and looking at the RNA versus protein levels for some of these regulators. So on the left, we have this is doesn't really matter which transcription factor it is, but this is the single cell RNA-seq measured mRNA levels, and then on the right is the protein levels for the same gene as measured by GFP in our imaging studies. We have hundreds of genes we can do these for. We always see the same pattern where there's this lag. You can see if you plot GFP versus or protein versus mRNA, you get a pretty poor correlation, similar to what Shalev was seeing in the intestine. And what's cool here is, and we're just starting to do this, now we have the tools to be able to actually not just learn the protein degradation rate and the translation rate, but actually to look for exceptions within particular lineages where it looks like there might be it's not well fit by a single model and there must be some kind of regulation. So I just want to close there, thanking people in my lab who have done really well, surviving from times we can be close together to times when we can't. Philadelphia is an amazing city. People who are interested in postdoctoral opportunities or in graduate school, highly recommend it. And I'll stop and take questions.
Thank you very much, John. We are a little over on time, but I think we can maybe squeeze in one question or two. So Christian Ray asks, well, first says cool work, and then says, do you have any idea about if tissue homeostasis or lack thereof in C. elegans may affect developmental trajectories or lineages?
Yeah, I'm not sure I totally understand the question. So tissue homeostasis. So what's interesting about C. elegans is like in most cases, there's very little sort of compensation for changes in neighboring cells. So for example, if you ablate a particular cell, the 16 cell stage, basically the cell face that would have come from that cell don't appear anymore. Whereas most of the other cells, they produce their normal face. So a few exceptions, late state inductions that happen. So basically we think that there's little of that. But we're quite interested actually, then there's interesting ideas we have for single cell approaches to try to test specifically for cell fates that require to basically, cell contact in the embryo to be induced. And I think we perhaps will identify more of those if we look systematically.
I think since we're a bit over on time, we will just move on to Katie. But thank you again, John. Katie, the floor is yours. Oh, I'm sorry. Introducing Katie Galloway from MIT, talking about detangling DNA, single-cell approaches to understanding plasticity in cellular reprogramming.
Yes. Hello. Thank you so much for having me. I'm delighted to be here, and I'm delighted for all of you who are here on this Friday night. I'm excited to bring you the story about how we were trying to understand sort of the fundamental limits of why cells reprogram and what undergirds plasticity. So reprogramming is, I think, really exciting for a lot of different reasons, especially because it provides the potential to think about fulfilling the promise of regenerative medicine by providing cellular therapies, creating cells for disease modeling, and ultimately for therapeutic screening. And so that's sort of the promise of where it's going. But the thing that I think is really scientifically interesting is the fact that we don't really understand this process or exactly all of the limits. And we've seen some lovely talks today thinking about how do we track this process? How do we drive it? How do we quantitatively characterize it? And I'm going to be telling you a little bit today about what we've seen in our specific system where we take fibroblasts, overexpress these transcription factors that make motor neurons, and can then monitor the process in this way. And the thing that got me really excited about this was it's just a fundamental conundrum that's hidden in this cartoon, but revealed when you actually look at the cells in a dish. Clearly you can see here that one of these cells, which these are labeled with HB9-GFP to make motor neurons, one of them looks like a neuron and one does not. One's really trying hard, but it just never really gets there. It's kind of stuck. It's got this arrested development. So this is the reprogramming enigma. We have essentially the same input. It's not a difference in viruses between these two cells, and yet they have these different outcomes. And so the question is why? Why do these cells make different choices? And what I think is, again, interesting about this, I think of this as this problem, at least how we approach this is a developmental synthetic biology, where we're trying to build these cellular transitions so we can understand plasticity. And when people look at this problem, I think depending on who you are, this is like a real Rorschach test. Like, what's the limitation? Well, if you're an epigeneticist, it's epigenetics. And if you're a synthetic biologist like I was, I'm like, oh, it's just the genetic programs, build one more sophisticated and it'll work. And neither of those are entirely wrong, but I think that there's a different view that actually allowed us to kind of advance on this problem and think about more of this, not from so much a memory problem, borrowing the analogy from computers like with epigenetics, or a software problem a la synthetic biology, but more as a processing problem, and specifically at the level of DNA. And so I'm going to give you all the talk in hopefully just one slide here. What we found was that there existed this rare population with high processing capacity. And by that I mean there were cells that could have high transcription rates combined with high proliferation rates. And that population was extremely rare. But if you could expand that population, and as we were able to do, we found that those cells would reprogram very robustly at high rates, and were also marked by signatures of maturity, both at the transcriptional level and functionally. And so look, the quest to understand this was then what are the mechanisms that actually undergird this process, and why is this process rare? How do we promote it? And so I'm going to tell you this is what we found, and I'm going to take you through how we got there, and then where I think it's leading us in terms of how we actually design genetic circuits to facilitate reprogramming and a whole lot of other cellular engineering strategies. So we got started with this work and even thinking about proliferation rate reprogramming from this paper from 2014, the Guo lab, where they noticed that fast cycling cells were this privileged state in reprogramming. And so if you separate them out based on CFSE, which is a dye that allows you to mark the fast cells based on dilution, so the dim ones are fast, you can see that those are the cells that preferentially reprogram and represent this privileged state. But it was a little bit challenging to figure out if proliferation was actually promoting reprogramming to a fast cycling state, or if it was just, fast cycling cells become fast cycling cells by being fast cycling cells. And so we kind of wanted to ask, well, is fast cycling something that actually promotes reprogramming to post-mitotic fate? And are there other determinants? And we actually did observe that fast cycling promoted reprogramming, but it wasn't deterministic. So it wasn't enough. If you make a fast cycling cell, it doesn't become a neuron right away. And so we asked, what's the other possible determinant? And we turned toward transcription. And so I'm so glad that John talked about how much transcriptional variability there is, because we thought, okay, if proliferation isn't the determinant, could it be the combination of transcription and proliferation that made these cells elite, that made them rare? So we developed an assay. This is a high throughput flow assay where we label our cells with CFSE a day after we add in our factors. And then we allow them to divide for 72 hours after that labeling. And then 4 days post-infection, we feed EU, which allows us then to label our transcripts, the RNA that's being synthesized with the EU incorporation to those transcripts. We then perform a click reaction that allows us to then measure the transcription rate based on the fluorescence. And then we can flow these cells to analyze them for proliferation and transcription. And the first thing we noticed was that our cells that were up here bright, our slow cycling cells had high transcription rates, and our fast cycling cells over here had low transcription rates. And that different genetic perturbations and chemical perturbations moved both the CFSC, so our proliferation, and our transcription rate. And the cells that we call these hypertranscribing, hyperproliferating cells were rare, and their transcription rate was high relative to their place as highly proliferative cells, but it wasn't high in the global scheme of things. And if we hadn't had this determinant of CFSC to observe them, we would entirely have missed them because they're right here in the middle. They kind of look average. They're just your average Joe, but they're punching above their weight for their proliferation status. And so we thought, could these be the cells? And what we noticed is as we expanded this population, we could see so we increased these HHC population with these combinations, and we could see a really huge increase in our reprogramming rate. It's not as compelling when you just see dots on a screen, but when you see the cells in a dish, it's pretty remarkable the difference we were able to achieve. We went from a handful of cells in a dish to really these nice, dense cultures. And ultimately, I won't have time to take you through this, but we did demonstrate that the HHCs are the elite population that preferentially reprograms through a series of somewhat sophisticated experiments that I won't take you through. But what we thought about, thinking more about the mechanism, was how are these cells able then to deal with these high rates of transcription and replication And what does that mean for us generally wanting to make an elite cell, a highly plastic cell that can do these things? And specifically, we thought if you have a high transcription rate and a high proliferation rate, you are going to be inducing a lot of stress on the DNA via the effects of DNA supercoiling introduced through replication and through transcription. And so if you don't, and you're going to need then enzymes to deal with that. And so we wanted to ask the question, do we actually see the stress that you would expect to observe? And indeed, what we found is that whether it's chromatin bridges or micronuclei, we saw that the introduction of these reprogramming factors induced a lot of genomic stress. Additionally, our reprogramming cocktail, which I'll just refer to as DDRR, a combination of P53, HRAS, and RepSox, a TGF-beta inhibitor, reduced this stress significantly and also limited the formation of these binucleated induced motor neurons, suggesting that it was somehow mitigating this genomic stress. So we wanted to know what are the molecular mechanisms that actually underpin this. What could it be that's labeling these that's allowing these HHCs to perform at these high processing functions? So we went in and performed a single cell RNA sequencing experiment across the reprogramming time. We took fibroblast hyperproliferative cells at day 4, HB9 GFP positive cells at day 8, and then the final induced motor neurons at day 14. Putting them on this landscape, we were able to identify a population of putative HHCs based on their proliferative status. We noticed that they were marked by topoisomerases. Topoisomerases came up as being an interesting hit, and it kind of started to check the boxes for us in terms of thinking about genomic stress. So topoisomerases resolve supercoils that are introduced by transcription and replication, and therefore they'd be important for reducing stress. We knock these down, you lose the HHCs, you lose your reprogramming rate. Additionally, if you treat them with inhibitors instead of shRNAs, similarly, you drop your reprogramming rate. And so these populations of HHCs are supported by these topoisomerases, which gave us some interesting ideas about what it might look like then to think about designing circuits that could actually promote reprogramming. And what I mean by that is we know then that these genomic processes are limiting our transcriptional activity in terms of our ability to induce the gene networks that we want to turn on in reprogramming. So we wanted to ask the question, can we see this at a single genetic locus using a circuit as a test bed system? And I'm going to skip through this because I'm running just a little bit behind. But what I want to tell you is that we took a circuit, a simple inducible circuit, knowing that you could put it into 2 different orientations, that these 2 different orientations would induce different levels of genomic entanglement, and then built a model to predict how that genomic entanglement would then manifest as different levels of gene expression. We could see that if in a tandem orientation, a circuit would have very little coupling, whereas our divergent and convergent orientations would start to interfere, suggesting that we could actually, at the level of chromatin, potentially engineer how these elements interact. And again, I'm running out of time, but the thing that I'd like to leave you with is we've got our first really exciting result in that our tandem orientation, where we have this reporter that then is co-expressed with this inducible element, we see that there's this correlation expression, which is exactly what you'd expect based off our model. There shouldn't be really any coupling or any drop. Whereas when we had our convergent orientation, we predict negative feedback through the coupling of the physical coupling of these 2 elements, and you see a drop. And that's exactly what we saw when we just simply flipped around. These are the exact logical exact logically same circuits, and yet their physical coupling is resulting in vastly different expression profiles. We think that's really interesting because this physical coupling should also be controlled by the expression of topoisomerases, potentially meaning our circuits can serve as sensors for these different populations. And if you have high expression of these topoisomerases that we see in HHCs, we'd expect that you'd have high activity, and they'd be able to untangle these circuits. That's what we predict. And we have some preliminary results that show that these populations where you have high expression of both parts of your circuit only exist in the case where you're not inhibiting these topoisomerase isomerases with the camptothecin, specifically top1, and we did not observe these results when we inhibited with top2A via doxorubicin. So we're really excited about this. We are enthusiastic to start thinking a lot more about how do we physically engineer circuits. Most of the work in synthetic biology is focused on the logical functions and the organization or dimerization. But I think the physical realization is something that is, it's the right time for us to start looking at this, and especially using the new genomics tools, including micro-C, to look at the structures. So with that, I would just like to conclude with this is all in support of our goal of actually building circuits that can support regenerative medicine. I'm grateful, especially to my lab, who has endured a very challenging year. I started in July 2019, and my students started in 2020 with me in January, and we've grown very rapidly. We're still recruiting, and I'd be glad if you could, for those who are interested in joining us. Also, this story started in the Acheta lab at USC, where I was a postdoc, and I'm thankful to Justin, and especially Kim Wonder, who's a graduate student on this project. With that, thank you for your time and attention, and I'm glad to take questions if we have time.
Thank you, Katie, for a wonderful talk. We've got time for a little off, but we can do a couple of questions. So King Sun asks, or says, super cool work. I want to ask, how precise is the reprogramming? And do hyperproliferative cells also give a higher risk to form cancer?
Yes. So how precise is the process? I mean, I think we're still only reprogramming. If you think about precision in terms of like how many cells can we reprogram in our dish, we can make about 33 or 1/3 of the cells be HHCs, and those seem to reprogram, highly efficiently. But I'd say it's still not that well controlled. And I think we can do better. That's the aim of a lot of what we're thinking about with these gene circuits, to control the reprogramming process. But we feel like the circuits themselves have to be well controlled and well behaved within the genetic environment. And that's not something in 3D we think is actually achievable yet. But we're mapping that out. And in terms of cancer, yes, absolutely. I think regeneration, plasticity, cancer development, they're all balanced on an edge of a knife. Anything that will allow you to transition in one direction is essentially the opportunity to transition into something that, I guess, to de-differentiate. And I think that always makes us wonder if you're headed towards cancer. So to be able to control those processes for regeneration, I think, is inherently balancing the risk with cancer. At least that's my personal thought on it.
Cool. We'll do one more question. So Geetika Arakatia asks, cool talk, are the elite cells always elite? Or did you happen to catch them at the right time? Basically, does every fibroblast have the potential to be elite at some point?
Ooh, I love this question. We ask this question all the time. Like, if you rolled the dice and seeded the cells a different way, would the same cells reprogram? Or is there some sort of ergodic behavior in which the cells would come back, and it just depends on when you would hit them? But the distribution itself is kind of fixed, but any given cell's potential is not. I tend to think that's the case, but I don't know if we have any evidence to support that. And I think that's even thinking about the experiment you would do to test that is, I think, a little bit complicated. With proliferation, it's obviously true. If you take the fast cycling cells and then seed them, they'll recapitulate the distribution that they came from. So in reprogramming, we have an arrow in time in which there's irreversibility. So it's hard to ask that question. But certainly with proliferation, it's true, if that's our only determinant.
Yeah, such a fascinating question. I don't know. I always kind of wonder about it. Anyway, thank you again, Katie, for a wonderful talk. And I will turn it over to John to introduce our next speaker.
Right. Up next, we have Michael Ratz from the Karolinska Institute.
Yeah. Hello, everyone. Thanks to the organizers for giving me the opportunity to speak. I'm Michael. I'm a postdoc in the lab of Jonas Friesen at KI in Stockholm, Sweden. And we are interested in, so our motivation comes from our interest in stem cells, and particularly we study a process called adult neurogenesis, where we very often use a tool which is based on in vivo clonal tracking, to identify the progeny of all progeny of a single cell. And while this is a very powerful tool, it really suffers from low throughput because we rely on sparse labeling, and it doesn't really provide any molecular information apart from a few markers that we can stain for with antibodies. But on the other hand, of course, we have technology like single-cell RNA sequencing, which is enabling us to profile thousands, even millions of cells in one assay, and basically at high throughput obtain whole transcriptome information. But often we don't have lineage information, and very often we also rely on dissociated cells. So as other speakers already introduced before, we're basically we want to solve that and basically combine the best of both worlds by introducing expressed genetic barcodes into brain cell progenitors that then can be captured later on using RNA sequencing. And I think these cellular barcoding approaches in neuroscience there can be very powerful, and I think there are like three very big areas where this can contribute to. I think what most people study is, of course, how diverse cell types are generated from a very homogeneous pool of progenitor cells. I think what's really interesting too is to relate gene expression to neuronal connectivity and ultimately activity and behavior. And I think what's also really interesting is to study how neuron lineage and birth timing influence ultimately wiring specificity. And I think lineage tracing or clonal tracking coupled to RNA-seq is very powerful in that area because we don't really have a good understanding of the molecular mechanisms underlying this. So our contribution to this area is an approach that we call TRACS, and this stands for clonal tracing and expression profiling in the mouse brain using single-cell RNA-seq, and relies on a lentivirus library similar to what Klein described or SMF Morris also was working on. It's basically a lentivirus that encodes a nuclear localized EGFP. In the UTR, you have a barcode, which we call a clonelD, and this is upstream of a polyA site for single-cell RNA-seq capture. And the way we barcode the cells is we use a ultrasound-guided in utero injection. So we're interested in the developing mouse brain. So we inject roughly 1 million barcodes into the mouse forebrain at embryonic time point E9.5. That's just before neurogenesis begins. And we think that under our experimental conditions, we have most of the cells uniquely labeled with only single clonelD. Following our injection, we wait about 3 weeks and then isolate cells from the 2-week-old mouse brain, where we then isolate barcoded cells using FACS from 3 different areas. And we use droplet-based single-cell RNA-seq to reveal the transcriptomes and also the barcodes. And here's the entire data set, which is corresponding to 4 brains and a single control. We have around 62,000 cells, many diverse cell types. So there's 40 different cell types coming from these different regions, which is the cortex, the striatum, and hippocampus. So these are forebrain regions of the mouse brain. And we see that there are some cells which are, of course, found in all 3 regions, but many cells are also region-specific. For example, glutamatergic excitatory neurons we mostly find in the cortex, or radially-like stem cells, mostly in the hippocampus. And because we're the non-injected controls, we first assessed cell health just to see whether lentiviral barcoding somehow messes with the cell physiology. And we first correlate average gene expression between classes of cells of barcoded and non-injected controls. We see there's a very high correlation. And we also basically investigate whether we have certain immune response genes that are associated with an HIV infection, which the lentiviral barcode is based on, or the lentivirus genome. And we can see an obvious upregulation of such an immune response genes. Genes that we see whether differentially expressed between barcoded and non-injected cells are, for example, EGFP, which of course makes total sense because it's a transgene that we introduce. So from that, we subset our data on only our barcoded cells, which are roughly 50,000. We extract clonelDs from the data, get from something like 19,000 cells, we get clonelD information. Most of the cells have like a single clonelD, something like 10% contain 2 or more. And then we can now use this data to reconstruct clones. And overall, it's about it's more than 2,000 clones and 12,000 cells in these clones. And I just want to point out here that if you would want to collect data of 2,000 clones using classical clonal tracing, you would definitely end up with sacrificing hundreds of mice, because you really rely on sparse labeling. We find that 25% of our total population of cells are containing clones. Since we have so many different subtypes of cells, this of course varies across cell types. But I think what's one interesting aspect is to look at the general clone size. And we saw here like a dramatic difference between cell types that originate from, that are mesoderm derived, like D cells. These are microglia and perivascular macrophages. And they have like roughly 8 times bigger, they make 8 times bigger clones than neuroectoderm derived cells, such as neurons or glia cells. And we think this makes sense because there's only a small window where these cells basically migrate into the brain. So you have a very small population of progenitors that then probably needs to increase the clone size rapidly to then have enough cells to populate the entire brain. Another aspect we looked at is the regional distribution of these barcodes, these clonelDs, and we find that most of them they accumulate in one of the 3 regions. And if you find clones that are regionally dispersed, this dispersion often happens between the region of the cortex and the striatum, or cortex and hippocampus. And now we zoom in a little bit into the cell types that are associated with these more diverse or dispersed clones. And that's basically quantified here, where we have the percentage of cells that are spread across more than one region. I just would like to highlight 2 examples here. So one region that is barely crossed is between the striatum and the hippocampus, and we find clones that do that, and they are most of the time associated with oligodendrocytes. So these seem to be very migratory cells that we know are generated in this part, in the ventral area of the brain, and then they seem to kind of migrate out and populate other parts of the mouse forebrain. And what we also came across again were, again, microglia. These seem to be the only set types that are very migratory. So they don't only produce huge clones, they also are very migratory cells that seem to enter the brain in one location, and then from there kind of spread out over the entire tissue. Another aspect that we were interested to look at in the data is the fate distribution of clonelDs, and here we use a tool that was also introduced by Klein in this science paper in 2018, and we calculate what we call the clonal coupling score, which is simply the number of shared clonelDs relative to randomized data. And when you do that, you see that you have in that heat map, you basically get 4 groups or 4 clusters of cells, and they mostly cluster by region. And I would like to draw attention to this hippocampus cluster, which itself is containing 2 smaller subclusters. And here we think we are talking about 2 different progenitors, fates, and fate 1 is a progenitor cell that mainly makes astrocytes and neurons, while there's a second fate that is already biased at a very early stage, which is E9.5, just before neurogenesis begins, that just makes astropanel cells and oligodendrocytes. And finally, as I told you, we would like to correlate the transcriptome also to the connectome, or even read out neuronal connectivity using transcriptomics. And for this, we use, of course, in situ sequencing, and particularly we use spatial transcriptomics. And our approach that we call space tracks, because it's transcription expression profiling in situ or in space, allows us to read out clonality, cell type, and transcriptome. So we take a barcoded brain, make tissue sections, we mount them on a glass slide, fix them, and do an immunostaining step, which allows us to essentially reveal the cell type, the broad cell type of barcoded cells. And this is how the data looks like. We, as I said, from immunostaining, we can distinguish between major cell types. In our case, it's neuron oligodendrocytes. We can reconstruct clones, and interestingly, they're significantly larger than when you use the tracks approach, which relies on dissociated cells. And that's probably because we, of course, don't dissociate the cells, and this leads to reduced cell loss. Overall, we see a similar pattern that we have an accumulation of clonelDs in major brain regions, but now we can also zoom in and see that there's like large dispersions within these regions and information that we couldn't assess before. And finally, we can also reconstruct kind of classical clonal migration patterns that were described previously in the mouse brain using this technology. So here you have dispersed clones, they're far spread over the tissue, and you have also these clusters like radially oriented clones in the cortex. And as an outlook, I would just briefly mention that we can actually also study this, use this technology to study the clonal maintenance of gene expression. So we ask how clonally related cells, or how similar they are, and whether they maybe share heritable gene expression states. So we basically look at gene expression similarities beyond cell types. And I'm pretty excited about this data because we see, for example, that one of these clonally expressed genes is the transgene that we actually put into our cells. So I consider this a positive control because each clone shown here in the violin plot basically is represented by one progenitor cell. So it makes sense that between progenitors we have slightly different clone and EGFP expression levels because the transgene is integrated into different loci. And another gene that we find in these radioglial-like stem cells in the hippocampus is, for example, claudin, which is a component of tight junctions. And a final example is a molecule called the cellular molecule 2, which we see in, say, one glutamatergic neurons in the hippocampus, and which seems to be kind of clonally maintained between related cells. So in summary, I showed you a technology that we use for retrospective clonal tracking and expression profiling in the mouse brain. After a one-time in vivo barcoding step, we identify unique features of brain macrophages such that they make these ultra-large clones that are regionally dispersed, and these cells basically have a single progenitor origin. We see already very early, just before neurogenesis, two populations of fate-biased progenitors at the hippocampus. And we introduce a technique we call space tracks to simultaneously do clonal tracking and expression profiling in situ. And we also try to identify first kind of genes that are associated with clonal gene expression. And this brings me to the end of my talk. These are the people that contributed to our work. That's our lab in Stockholm. And thanks for your attention.
Thanks, Michael. We have a question from Maria Luisa Sequeira Lopez. It says, fascinating approaches and data. I agree. Do you see any developmental or iatrogenic effects in postnatal life, including seizures due to direct injection in the embryonic brain?
Yeah, I can't say. I wouldn't expect it because I think it's because we don't track the animals until they're actually adults. So we usually sacrifice them after 2 or 3 weeks after birth. But I wouldn't suspect it because I think these neutron injections are very widely used. Other people use it for much longer time points, and I wouldn't expect any detrimental effects from that point of view.
Maybe I'll ask a question as well. It's also sort of a technical one. I'm curious, to what extent does the accessibility of cells to the injected material bias what cells are available for you to be able to track using this method?
Yeah, that's a good question. So right now we do this intraventricular injections. So basically into the lumen of the ventricle, which is a volume in the developing brain, and we mainly target cells that are, of course, facing this ventricular volume and the ventricular zone. For us, that's great because these are the actual stem cells that we're interested in. I think at later time points, it becomes a bit more difficult because this volume becomes very tight. But what we're also doing, we're working, for example, with viruses like AAVs that spread more within the tissue and that can actually also be coupled with transgenic lines to target other cell populations.
So I think we have to move on to our last speaker. I encourage people to look in the forum to answer any other remaining questions. The last speaker of the session and the conference, I guess, is Merit Romaika from Max Britz Labs in Vienna.
Yes, thank you so much. I had a really good time the last couple of days, so thanks for putting together this wonderful conference. And I'm super excited to share this work today with you. So my name is Merit Romaika. I'm a PhD student in the lab of Christa Bücker, and we are interested in really one fundamental question. So really, how can a cell leave their cell state and how to then enter the next one? And the model system we're using for this is the exit from naive pluripotency. So this is also cool because in an embryo in vivo, this happens while the embryo is implanting into the uterus. So it's super hard to study in vivo, but we're using an in vitro system for this. So we keep our mouse embryonic stem cells under defined culture conditions. In these culture conditions, they are very stable, they can self-renew, but as soon as we exchange these culture conditions, cells are committing to formative pluripotency. Roughly 24 hours into this process, they reach a point of no return. So if you then would put them back into the original conditions, they are not able to self-renew. But if you just let them go within 48 hours, they have reached a state of formative pluripotency. So why are we talking about these two different flavors of pluripotency? So the core pluripotency network is shared between the two states, but these factors are interacting with different interaction partners and therefore also have different chromatin binding patterns. We see a change in the transcriptional landscape and in the enhancer landscape. This transition is very much regulated. So there are 5 major signaling pathways involved, and we heard a lot about ERK signaling before. But what is now striking is you really have to hit 3 of these pathways at the same time to completely block differentiation. And if you're not doing this complete hit, so if you just hit one of the factors, we see some kind of problem in differentiation. So they are somehow impaired, but this is exactly what we want to study. How can such a cell fate transition, even though it's impaired, can be executed? So we decided to look at 2 different mutants, so always single mutants at this time. So for one, we take the TCF7L1 knockout. This is downstream of Wnt beta-catenin signaling. Normally, TCF7L1 is repressed by beta-catenin, but if beta-catenin is removed, TCF7L1 itself can become active and then works as a repressor on the naive pluripotency network. And as another factor, we are also studying RBPJ. This is a downstream effector of the Notch signaling pathway. So what do I mean with differentiation impaired? If we look at this transition just with bulk RNA sequencing, you can see here that these control cells, so the dark blue ones, are committing a lot of transcriptional changes, even within 16 hours. But it's also clear that these mutants are not able to commit to the same changes in there. And of course, now we can do a lot of differential gene expression, but that doesn't really help us to find out why they are showing this phenotype. Because if we start comparing these cell states, we will see a lot of indirect effects just because the cells are really not where they should be. They're just not in the right cell state. And to solve this problem, we then turn to single cell sequencing. So first of all, I looked at my control cells. So all of these cells are control cells. And I multiplexed different time points and then later on also different genotypes by multiseq. This really helps us to avoid any batch effects and to have unconfounded data in there. So if you look now at this UMAP, you can really see that we are sampling the continuum of cell states. And I should say in this context that if I say cell state, I don't mean a stable attractor cell, but I really mean this intermediate microstates in there. So it's really one cell fate transition. So we have one start point, one end point, no branching points, one uniform direction in there. So this is also what we see if we now sort the cells in pseudotime. So we see this constant progression of the experimental time points. And on a technical note, I have some cells where I just couldn't find my multi-seq barcode anymore, so I don't have the time point information. But we decided to leave the cells inside because for us, it showed that this is really a random sampling and these cells are able to commit to all strangers, so they sample the whole space in there. Now, it's a super, or it seems to be a super straightforward system, but even if you look here at this 12-hour time points, we already see this huge spread in the cells. So they're really, even though they're collected at the same time, they're sampling different cell states. And it's super interesting why we're doing this, but this is part of another story, and the cell cycle is one hot contender for this differentiation differences. So all of this now was the control situation. What happens if we start to break things? What happens if we look at the mutants? So I did exactly the same experiment, also in parallel to, again, avoid batch effects. And if we now sort these cells, you see these differences showing up. So if I'm just looking at these populations, they're really not able to commit to the same changes, and these cells are not in the same cell state anymore. This effect is even stronger if we look at the TCF7L1 mutant. So if you check here, these later time points, there is no progression at all. So the peaks are not shifting. On a side note, and very important, we do see the shoulder of cells appearing. So there are some cells which are escaping whatever problem they have. And this also fits with our expectation because we're not completely abolishing this differentiation. We're just impairing. Eventually, these cells will always differentiate. But what you can clearly see from this, even if we now take fine-tuned time points, we are comparing different cell states with each other. And this always is a problem because then we will also see these indirect effects. And this is something we see on expression level. So for example, what we like to do is use FACS reporters for this transition. So I have GFP under control of a naive marker. That means that for control cells, we see downregulation of GFP and we can monitor the transition like this. And you can see that the TCF7L1 knockouts are not able to downregulate this marker at the same speed. Similar, if I now look at my single-cell transcriptional data, but bin it back to the experimental time points, the control cells are able to downregulate, but this mutant is not able to downregulate REX1. But here we always have this issue now that we have different cell states. And now we can start correcting for this. So now I'm taking each cell and I'm placing each cell in the x-axis according to their position on pseudo-time. And if we do this now, you see that we lose this difference, which would mean that before we really just had this indirect effects there. Importantly, there are a couple of cells of the TCF7L1 mutants which do make it to this later points in pseudo-time. And these cells are also able to downregulate REX1. This also matches, if you then look closely at this FACS plot, we do have some cells which are able to show this differentiation. And this is not just one marker gene we're looking at, but we can also take this now to different cells. So I'm showing you another marker gene for a naive gene, which is downregulated in this process. And we can correct for this downregulation and also for genes which are upregulated. So this is a formative marker gene. Again, we can correct for indirect effects. But very importantly for us, we are not just overcorrecting any differences, but we're correcting for cell state, and what we should be left with should be the direct effects. And for the sake of time, I'm just showing you one gene where this is happening. So KLF2 has been described before as being associated to the naive network, so the starter network. But very importantly, it can be decoupled from the cell state transition. And even more important in our context, TCF7L1 is directly repressing KLF2. And this is something we can recapitulate in this differentiation. So by correcting for the cell state differences, we can correct indirect effects. And what we're left over with are these direct effects. Now, we can also take it now to the next step and take it away from cells and take it really to a function of gene regulation in pseudo-time. And I really have to say the TradeSeq package and the newer developments of this package were fundamental in doing this kind of analysis. So what I'm showing now are not cells anymore, but really positions in pseudo-time. And what you could see for the control cells and for the RBPJ knockout cells, that they show the downregulation what we expect. But in the TCF7L1 knockouts, there's this escape on this gene. And because TradeSeq can now test for not only different gene expression in pseudo-time, but also different dynamics and kinetics of gene expression of pseudo-time, we can now really take this with our corrected cell states and apply it to this indirect versus direct effects. So to sum up, if we look at time points, we will always have the differences in cell state, and it seems to be that these differences are really driving the analysis. But when we now perform the single cell time courses, we can first of all describe this difference in cell state, and then we're able to correct for these differences. And with these corrections, we can now compensate indirect effects, and we're left with the direct effects. And what we're doing right now is take this compensated cell states, and now we want to build the dynamic gene regulatory networks which are governing these changes. And with this, I'm left to acknowledge all the people who were super supportive in this work. First and foremost, my supervisor, Krista, and I'm happy to take any questions.
Great talk. Yeah, so it's fascinating that you can see this decoupling of time and the slower progression along pseudotime in addition to the more specific defects. And what do you think is the cause for that? Do you have any does the network predict that, or are there specific regulators you think that are actually controlling that progression of that?
I think that was a theme which was coming up earlier today. So we don't believe that there's this major driving force. It's not one regulator, but it's really that these cells are integrating different pathways. And then the question is now really, so if you have an additional effect, so little effects from these different pathways, it's probably that you're just not as able to directly change when the cues are coming. So we do think these are smaller, subtle effects, which then add on later.
Great.
I guess I have one quick question, which is, given that it is sort of multifactorial or dependent on a lot of, I guess it's maybe a bigger picture question. How are we going to control or engineer such processes if they're controlled by so many factors that we can't possibly control them all? What might be strategies that we could employ?
Yeah, especially because it's so multifactorial, you probably don't get it to it while just taking out one factor after the other. So yeah, if you want to build it up from ground on, I think that's super challenging. But also the fascination is because it is integrating so many different levels, and eventually these cells differentiate. So eventually they can deal with these challenges.
So we have a question from D who, given I guess the sort of differences in pluripotency network or in between mouse and humans, do you think that there would be sort of a similar story in both, or do you think there's something different?
Yeah, one interesting side note is KLF2. So the direct factor, which I showed, is thought to be a mouse-specific addition to the naive network. So it's probably the exception to the rule. That being said, it's also that this naive state really is more stabilized in the mouse system, not so much in the human system. So in the human system, you really have to try to keep human stem cells as naive cells. But in general, the state is there, it's just not as stable.
Great. Well, I'd like to take the opportunity just to thank all the other speakers and for a wonderful session. And I'm going to hand it over to Arjun for some closing remarks.
Yeah, thank you, John, and thank you to all of the session speakers. Just a fascinating session. And basically, thanks to everybody for making this just such a wonderful conference. I can say that I learned an incredible amount over these few days, and I think it really captured the conference really captured the energy of what I think the next phase of the single cell field is going to be, which are a lot of biological applications. I think what I really want to do is thank a lot of people for making this happen. Especially, I want to thank my co-chair Shalev Itzkovitz, who unfortunately it's very late in Israel right now. Shalev just really did an incredible job putting together this program, and it's been a joy to work with him. He retains his title as nicest person in science for me. I want to thank all the speakers who signed up to give yet another Zoom talk. And I, want to thank, of course, all the attendees who really made the conference a living, breathing thing. Hang on. I also just really want to thank, huge thanks to all the Keystone staff, Allison, Shannon, Lindsay, Josh, Nick, Grayson, Dale, just everybody for making this an incredibly smooth thing. I can tell you that both Shala were really scared when we started. Like, how are we going to do this? Like, I don't know how all this tech stuff works, but they really made this as smooth as we could have possibly imagined. And they really made this virtual experience just, I think, as good as it could possibly be. Obviously, it's not quite Banff, which, obviously has skiing and all kinds of fun things. But there were, I think, some real positives to the virtual conference experience. And I think this comes from some of the numbers that we had. So we had 578 plus participants. And these participants came from over 40 countries. And that's really, I think, many people would probably have a hard time getting to, ski country in Canada. I think we got a lot of really great attendance, which was really wonderful to see. I think we also got a lot of really democratic participation. I think we got, 40 chat participants, many chat messages going through. The most chatty was Giovanni Goizian. So you win most chatty. Lots of questions submitted, and I feel like that was really democratized. Like we had questions from people around the world, 286 questions in the Q&A session. Most inquisitive, the title of most inquisitive goes to Zhu Shu with a full 9 questions submitted. And we even got a song out of this. So Uri gave a really great song about inflammation, which you can find on YouTube. And just to close us out, I think Shalev and I, as we were trying to think about what this conference might look like, I think, we've all been talking about transitions and transition states. I think the field itself is in a transition state between, the enormous technical growth that we've had in the last, really decade. And even before that, and now I think we're in a transition where we're really seeing the fruits of what the application of these technologies to novel biological systems can give us. So I'm just really excited to see how these conferences, what exciting science we're going to see from these same conferences in the coming years. And again, thank you all for participating. And yeah, hope to see you again soon sometime.